attend. We're really delighted to have both those of you on Zoom as well as in person, and to have Dr. Kiri Ness here today. So Kiri's well familiar with Minnesota. She's a physical therapist by training and then received her MPH and PhD in clinical epidemiology here at the University of Minnesota, where she's received some distinguished alumni awards, including last year for all of her work. She's a member in the Department of Epidemiology and Cancer Control at St. Jude and is the principal investigator of the Human Performance Lab there. She's probably best known for her roles with the executive committee with the Childhood Cancer Survivor Study, as well as with the the St. Jude Lifetime Cohort. And we're excited to hear about her work today, talking about aging and survivors of childhood cancers. And I know personally we're thrilled to have you here. So thanks for being here.
Thank you, Anne, for having me. And I said to Joe this morning, who else would you invite in February but somebody from Minnesota? Because, but the weather is super nice. So I'm going to talk a little bit about preventing and remediating accelerated aging in survivors of childhood cancer. So first, I'm going to just describe ways that aging and frailty are characterized in survivors. And then I'm going to talk about potential interventions to remediate frailty in survivors who are already frail. And then I'm going to conclude with talking about some exciting interventions that have the potential to prevent frailty in young survivors and in current patients. So frailty can be measured in cancer survivors or characterized in three different ways. First, on a global level as an accumulation of deficits or diseases. Second, on a phenotypic level using pre-specified criteria that characterize physiological health, where three or more of muscle wasting, muscle weakness, slow walking speed, fatigue, or low energy expenditure is considered a frail state, and frailty on a more mechanistic level using measures that signify molecular aging. So these, pretty much somebody who's frail will have deficits in each of these areas. So often they have multiple chronic diseases or multiple emotional, psychological, and physical health problems that interfere with daily function, or they might look physiologically bad. They look like an old person, but they're not really an old person. They're a young person that looks like an old person. When I first read about the Freed frailty criteria as a PT back in the day, it resonated with me. And I remember the first time that I really observed it, I was doing a study with Joe and we were looking at brain tumor survivors, and I was like, what the heck is going on here? Well, they're brain tumor survivors, so they look like little old people. But then I moved to St. Jude 18 years ago and I started seeing patients in my lab, and I was like, wait a minute, this isn't limited to the brain tumor survivors. They all look like old people. And so even though we talk about muscle wasting, muscle weakness, slow walking speed, fatigue, and low energy expenditure, there's investigators out there in the world who do things like look at facial aging. So they take photographs and they can identify somebody's biological age by just taking a photograph of their face. And if you can think about it as, having more wrinkles, and I'm interested in talking to the guy who I heard give that talk now, that there's a lot of cosmetic work that goes on, like what happens when people get a lot of Botox to their face, and does that change the way that he does this facial recognition? But it's super interesting. And then of course, there's some really good recent evidence on cellular aging that corresponds to these phenotypes and is providing us with some senescent drugs that we can do to treat maybe or prevent aging. So for those of you who don't know, the Childhood Cancer Survivor Study is a multi-institutional cohort of 31 institutions that was formulated on the back of a napkin in a bar. And it originated at the University of Minnesota. It's been continuously funded since 1994. It's a retrospective cohort of individuals diagnosed between 1970 and 1999 who survived 5 years from leukemia, lymphoma, CNS bone, Wilms tumor, neuroblastoma, soft tissue, and bone sarcomas. The uniqueness of this cohort at its inception was the detailed treatment data that was abstracted from the medical record and the foresight of the investigators to measure a wide range of outcomes. So not just chronic disease, but also psychosocial health and quality of life. So to talk about deficit accumulation, there's really nowhere better to start than to summarize disease burden in the childhood cancer survivor study. These data show that childhood cancer survivors have high rates of chronic disease in young adulthood, with the cumulative incidence of a first grade 3 to 5 chronic condition rising with time since diagnosis. So even among survivors treated more recently, the 20-year cumulative incidence of at least one grade 3 to 5 chronic condition is still 27.5%. Remember, these are young people. As a comparison, only 4.6% of the siblings experienced a chronic condition in the same time period. So CCSS data describing disease accumulation are supported by data from our St. Jude Lifetime cohort. And this cohort was actually assembled to facilitate longitudinal clinical evaluation of health outcomes in aging adults surviving pediatric cancer. So what's unique about the St. Jude Lifetime cohort is we see these people in person. They come to campus for 3 to 5 days every 5 years when they're adults and have a comprehensive battery of clinical evaluation. And prior to COVID, it was super unique because people just didn't really get clinical evaluations. Post-COVID, now we have figured out how to do clinical assessment using telehealth. So the CCSS cohort is currently having a group of people clinically assessed for accelerated aging over a Zoom platform. Actually, we use Teams for that, which is HIPAA compliant. So this cohort includes persons who were treated for cancer at St. Jude Children's Research Hospital, again, who survived 5 years following a diagnosis of childhood cancer. So there's 6,005 survivors who have been enrolled and had an in-person visit. This was right before COVID that we froze the data. We're currently freezing it again. And 773 controls. And the controls were recruited from friends and family members of current cancer patients so that they would not be all from the Memphis area. So almost all of them have had more than 2 evaluations now. And participation is 80.3%, which is typically unheard of, but we fly them in and we reimburse them. And they're devoted to Melissa Hudson, who's the PI, and because she was their physician. And about 90% of those who are contacted agree to come. There's some people we can't find. So the cohort's just over half male, 17% identify their race as Black, and 4% their ethnicity as Hispanic. Predominant tumor types at diagnosis are ALL, central nervous system tumors, and Hodgkin lymphoma, although many other tumor types are represented. And this cohort's not limited to the 8 diagnoses that were included in the childhood cancer survivor study, but includes other diagnoses as well. The burden or accumulation of chronic disease in the St. Jude Lifetime cohort is substantial, with the mean number of grade 3 to 5 chronic conditions by age 50 years at 5 per person. And that's a value twice that of community controls. And I think that it's higher in St. Jude Life because we see them clinically. So we're able to ascertain subclinical disease or asymptomatic disease kind of before it manifests itself and brings them to see their physician. So another way to describe aging in childhood cancer survivors is this phenotypic description. Several years ago, driven by the data suggesting accelerated aging from St. Jude Life, from our brain tumor study, and from CCSS, we looked at the aging literature to see if we might be able to identify an underlying phenotype. So to do this, we used performance measures and some self-report data and applied a phenotype from the aging literature, because in our observation of these brain tumor survivors who were in their 20s, they looked like they were in their 70s. So in the geriatric literature, physiologic frailty is a loss of physiologic capacity that interferes with normal function. It's described in older adults, and it is distinguishable from disability and comorbidity. It identifies individuals highly vulnerable to adverse health outcomes like hospitalization. It often precedes having a chronic disease, and it's an early predictor of mortality. We hypothesized that children with cancer experience early and then accelerated physiologic decline related to their disease and treatment exposure. So they get blasted with chemotherapy and other DNA damaging agents, or they might have a surgery that moves part of their brain or one of their limbs. And this is represented here by the dotted line. So if this is the case, one of our goals is to develop interventions that change the trajectory of this line. So frailty was first defined by Linda Fried in the cardiovascular health study among 65 to 101-year-olds as 3 or more of the following components of fitness: muscle wasting, muscle weakness, self-reported exhaustion, slow walking speed, and low energy expenditure. And those having 3 components or more were frail and 2 pre-frail. So in an initial analysis, frailty in the St. Jude lifetime cohort among survivors a mean age of 33 years was prevalent among nearly 8% of survivors, compared to a rate in the 65-plus-year-old cardiovascular health study of 6.2%. It was associated with a 2.6-fold increased risk of mortality among survivors over only 3 years of follow-up. To validate the findings and to get a larger cohort, we looked at frailty in the childhood cancer survivor study. The rates were slightly lower than those seen in the St. Jude lifetime cohort, likely because we had to rely on self-report. Nevertheless, rates were much higher than among siblings. So before designing an intervention, we wanted to see if frailty progressed with age. We wanted to know if this is something that happened at diagnosis or treatment, and then it just stayed static. So they started out low and they stayed low. But we invited 1,509 survivors back for a second St. Jude Life visit, and these survivors were 18 to 45 at their baseline assessment. Rates of frailty nearly doubled in 5 years. Baseline frailty was associated with a more than 3-fold increased risk of death in models that accounted for them already having a chronic disease. So to better understand what's driving this phenotype, this figure shows the prevalence of survivors who meet the criteria for each frailty component. So low lean mass, muscle weakness, and low energy expenditure are problematic, with muscle weakness and low energy expenditure substantially increasing in 5 years. And what's interesting and different about childhood cancer survivors than in older adults is that childhood cancer survivors appear to lose their muscle mass first and then function. In older adults, lose their function first because probably a neurological underpinning, and then they lose their mass. So it's sort of a similar but not exactly the same phenotype. So our approaches might need to be different. For instance, if you do an intervention with older adults, and I'll just draw on my way long time ago time when I worked as a PT in older adult populations, if you worked on their balance and their reaction time, you could usually improve function pretty rapidly, and you didn't have to have gains in strength. But in childhood cancer survivors, if you don't improve their strength, then you can't improve their function. So now I'm going to shift gears and show you some evidence of molecular aging in childhood cancer survivors. These data were generated by my colleagues, Xiaoming Wang, who's a computational biologist, and Mundira Kundu, who's an expert in essentially strange aging phenotypes in mice and mitochondrial disorders. And we have published, are in the process of publishing information about 2 primary hallmarks of aging and 2 antagonistic hallmarks of aging. So these figures show that telomere length is significantly shorter among survivors of childhood cancer than non-cancer controls, and that epigenetic age acceleration is also evident in childhood cancer survivors, with the slope of the line steeper among survivors compared to controls. So one of the things that we sort of noticed differently between telomere attrition and epigenetic age acceleration is that telomere attrition seems to happen early and stay fairly static, whereas epigenetic age acceleration appears to escalate. So one or both of these biomarkers might be a potential mechanism for, that we could target for future intervention studies designed to prevent or delay onset of chronic health conditions in pediatric cancer survivors. So as I showed a few slides ago, sarcopenia is a component of frailty and also prevalent in our cohort, represented by lean mass values lower than expected. So using correlative genotype data, when we look at mitochondrial DNA copy number, a 1 standard deviation decreases decrease in DNA copy number increases the odds of sarcopenia by 42%. So thus interventions that promote mitochondrial health. So, you know, we give a lot of mitochondrial health promoting drugs to transplant survivors to prevent GVHD. Might be something that, you know, intuitively was happening and we didn't know it and we were helping them. Or if we can prevent mitochondrial loss, it might benefit childhood cancer survivors. So although cellular senescence is largely undetectable in younger populations, when we evaluated P16 INK4A mitochondrial RNA expression in mRNA expression in peripheral blood T lymphocytes in a pilot study, this is 59 survivors, we found associations between expression and two potential markers of aging, walking speed and abnormal body composition. So one of my postdocs is really interested in this data, and she's published a recent paper showing that P16 levels are associated with cardiopulmonary function as well. And that was interesting to me. I sort of wasn't expecting that. So we'll do some more investigation in that area going forward. So now I'm going to talk about potential interventions to remediate frailty in survivors with multiple or specific deficit, phenotypic frailty, or with evidence of molecular aging. So first I'm going to talk about our intervention for prediabetes. This is a pilot intervention trial for diabetes prevention among adult survivors of childhood cancer that are in the St. Jude Lifetime cohort. And this is led by Dr. Stephanie Dixon, one of our physicians in the survivor clinic. And it's aimed as establishing the feasibility and preliminary efficacy of a combined metformin and lifestyle change intervention for diabetes prevention. So the survivors are enrolled to receive metformin and a digitally delivered lifestyle change program, that's NUM, over a 24-week period. And we measure glycemic control and insulin resistance at baseline, and then medication and adverse adherence and adverse events are obtained throughout the study. So our goal is to collect enough preliminary data to be able to do a larger trial to see if this is feasible. And I can tell you that what we're discovering in executing this trial and working with our NCI program officers is that survivors don't want to do the lifestyle intervention. No, I just want metformin. and so I think that it's been a really big learning experience. So NCI made us give everybody the lifestyle intervention first. Everybody has to do the lifestyle intervention. But they're annoyed by the platform. It's not so much that they don't want to talk to a dietitian or that they don't want to talk to a lifestyle coach, but they don't want to go log stuff. So that's really interesting, because in this digital age, they're like, I don't want to log yet another thing. I don't want to log stuff. I don't want to read all that stuff on my phone. I don't want to do it. Can you please just give me the medication? And these are not geriatric people. They're not like my generation, even, where, you know, like, if I need help with my cell phone, I ask an 8-year-old, tell me how to download this app. So I think that it's been a challenge to get people to want to do it. But now everybody's on metformin because most of them failed the lifestyle intervention and they're happy, and we're moving right along. And we'll have some good results to talk about, you know, whether or not we should just do a placebo-controlled trial and maybe add exercise that's supervised where people have contact with a human and instead of the app. So the lifestyle change is this NUM. This is commercially available. I'm sure you've heard of it. It's a smartphone-based lifestyle change platform. And the goal of their program is to develop sustainable, healthy lifestyle habits. And this was actually based on a lifestyle, their platform is based on the diabetes prevention program that the CDC developed. And it's the only one that's certified right now out there using the DPP. So that's why we picked this one, because these are pre-diabetic people. So there's a structured core curriculum and content, and it's progressive. And it talks about healthy eating, physical activity, and personalized goal setting, and self-monitoring. But remember, the DPP program was administered by DPP educators, not by a phone. And even though they have a chat GPT or a AI background on NUM that's supposed to like give you your own personal coach, I can tell you, and as a person who, since I'm doing a study, I sign up for the app, right? And then I make myself 5 different people. So I have 5 copies of the app. I have five me's that log on, and the different me's get the same information, even though the different me's are, I mean, unless Noom is smart enough to know that it's all me, but that could be, never know. So anyway, so this is a telehealth intervention in survivors with exercise intolerance, and it's called the Home Heart Health Study. And it's designed to test the efficacy of a telehealth-based intervention, individually tailored aerobic and strength training to improve functional capacity. And we are measuring cardiopulmonary fitness, musculoskeletal health, and neurosensory function. And then we also are measuring changes in methylation profiles from baseline to week 20 to see if we're impacting aging. So survivors have to have a peak VO2 less than 85% of predicted, and they also have to be younger than age 39 or 40 to be on the study. So this is our remote home-based delivery system supported by a Zoom interface. Hopefully this is gonna work. No. Let's see. There we go. You can't hear her, but this is Nicole talking. So she's demonstrating, you'll see the survivor on the other end of the Zoom. So the survivor is wearing a heart rate monitor. Nicole can see the heart rate monitor. And we just took some little clips, and now she's going to show you a strengthening. So this can be delivered at home. And again, this is at the beginning of the study when I first clipped this video, but I love this survivor, so I wanted to take a video of her. But you know, Nicole's demonstrating this in a not super nice gymmy type place, and that's very intentional so that the survivor can feel like they can do it in their home. And as I was telling the group last night, the survivors are allowed to pick their interventionist, just like Better Health. I looked it up, Better Health, where if you don't like your interventionist, you can pick a new interventionist. So this app, this can be delivered on a phone or a tablet or computer. We give all of our survivors who are in this study an iPad, and that's part of their reward. They get to keep the iPad at the end. So when the study is done, after 10 months, main part of the study is 16 weeks, but we follow them for 10 months. At 10 months, we disconnect ourselves from the iPad. They get to keep the app, but we disconnect us and we disconnect St. Jude. So then they can use their iPad. Like right now, because it's St. Jude, they can't download TikTok onto their iPad because we have a no TikTok rule. So we give them the iPad at the end, and our St. Jude IT can remotely release the iPad, so then it belongs to them. So that's one of the rewards they get. They also get, some other financial reimbursement. So, but it can be delivered on a phone too, and they can download the app on their phone if they want. And so I didn't put pictures of all of my staff up here, I should have, but what I do is when they get enrolled on the study and have their baseline assessment, then whoever's the assessor, that's a limited group of people, will then bring up the profiles of all the potential people who are available to be their interventionist. And they can read about it, and then they can click on who they want. And then that person is the one who contacts them. So we have a Spanish-speaking interventionist, 2 Spanish-speaking, 1 French-speaking interventionist for people who don't speak English as their primary language. Although my French-speaking interventionist hasn't had anybody speak French yet, but she's had people pick her. And then usually the kids pick her. I don't know why, but anyway, and then for one of our other studies. And then I have people who are African-American, just so people can pick somebody who looks like them. So this is another intervention, and it's been completed, and we did protein supplementation and resistance training, and it was designed to remediate frailty in survivors with low lean muscle mass. So the aims were to evaluate the effects of 24 weeks of supervised resistance training with or without protein supplementation on muscle mass, strength, walking speed, self-reported exhaustion, and physical activity. We also evaluated the interventional markers of cardiometabolic health, and so they were randomized to receive either resistance training in a placebo drink or resistance training in a protein supplement. So they were isocalorically the same amount of calories, not really a ton. And it had 21 grams of wheat protein per day, which I'm guessing was not enough because we didn't see any differences in change in lean mass between groups. However, both groups got stronger because we resistance trained them in person. So they were all from within 100 miles of the Memphis area, and the exercise scientists drove to the gym. We got them a gym membership closest to their home, and the exercise science person drove to the gym and exercised with them 3 times a week for 24 weeks. So it was expensive, mostly to pay the exercise scientists to drive all over the place. But we had really good adherence because, he and she, there was 2 of them, made appointments with them and kind of gave them a hard time if they didn't show up. I mean, people sometimes didn't show up, and we had interesting adverse events on that study, mostly related to people not being able to drive very well. So it appears that survivors respond well to strength training, but the addition of the protein supplement didn't enhance the intervention. So I've been doing lots of reading about proteins now and trying to decide if I can do a better job with a protein supplement. And also I have to work with this group of survivors who were in this study to figure out if we can deliver this at home, if they can lift heavy enough weights at home, because in the gym you can lift heavy weights and you have a spotter, but at home for safety, you're not going to have people doing a bench press with no spotter with heavy weights. So it will be interesting to see if I can figure out how to do this at home. So we've also have funding for this pharmaceutical nutraceutical study to address cellular senescence. And this study is a 2-arm, either dasatinib plus quercetin or a facetin alone, randomized open label intervention trial to improve cellular senescence, so P16 and walking speed, in survivors who are frail and have measurable P16 burden. And that's determined at baseline in 60 days within each individual arm. And this is to provide preliminary data to see if we can do a larger trial. So you realize that most young adults don't have any detectable P16. And that hasn't been our issue here, but one of the issues is even though they have detectable P16, there's some ceiling effects on a walking speed test in people who are in their 20s and 30s, even if they're frail, right? They still do better than a geriatric person because they don't have neurological problems. They have no muscle. So that's been another learning experience. And then here's my foray into the adult breast cancer world. So this is a study I'm doing with Mina Cedric at UCLA, and he's looking at he just got funded an R01 to look at the association or the effects of exercise plus facetin on frailty in postmenopausal women with stage 1 to 3 breast cancer treated with neoadjuvant chemo. They're enrolled within 12 months post-chemo, and they have to have diminished physical function based on their 6-minute walk test, 400 to 480 meters. So we haven't started yet, but we're getting ready to start. He got funded and then he moved to UCLA. So we're doing paperwork. So I'll be interested to see, this is what I'm most interested in. Which physiologists on my team are the little old ladies gonna pick? Actually, they're not little old. I mean, I asked Mina, how old are these people? He said, well, most of them aren't like 80. He said, they're gonna be like between 50 and 70, most of them, So those are interventions after frailty develops. So now I'm going to talk about interventions to prevent frailty. We don't want to wait till survivors are frail to do something about it. In the next series of slides, I'm going to talk about some interventions soon after therapy or during treatment that might prevent frailty. So the first one is a rewards-based physical activity. I'm in year 7 of this COG study because we had 2 years of COVID that interrupted it. But the primary aim was to find out if social interaction and rewards help improve fitness measured by the physiologic cost index. So that's, beats per meter, heartbeats per meter in the 6-minute walk test. It's an 18-month-long study. They have 4 clinic visits every 6 months, and the intervention period is during the first 6 months. So there's 2 groups. The control group, there's a little app. They can see their own activity. They wear a little monitor on their wrist, like a Fitbit kind of, but it's not a Fitbit. I'll show you what it is. And then the intervention group can see their own and others' activities and interact with others and earn rewards. So in the pilot, we figured out that if we give everybody rewards and everybody can see activity, that everybody will improve their function. So we wanted to see if you take away the interaction and they just see their activity, like your usual thing when you don't interact with anybody on your app, does it work? So they're 8 to 15-year-olds, all cancer cases with an ICD-10 code of 2 or 3 in remission and completed therapy in the last 12 months. They can't be too physically active, so they can't be on the swim team or, you know, really, really physically active. And some kids are pretty active. They have to be able to participate and they have to be able to use a computer. So this device is called the MoveBand 5. It was designed for children with developmental disabilities for the Special Olympics. So it's like $26 a device, so it's really financially reasonable. And the app is called Engage. The fitness tracker MoveBand 5, wears on a wrist, holds 3 days worth of data, has a rechargeable battery. As you can see here, you just plug it into the wall like any other thing. It's fully waterproof. The wristband's latex free and it's one size, fits most kids. And the data wirelessly syncs to their phone using Bluetooth, so you don't have to ask them to download it. It'll just sync. All they have to do is shake their wrist and it uploads. So sometime during the day, they're shaking their wrist and it uploads to the phone. If they didn't have a phone on this study, then we worked with their school to give them a little dongle for the computer at school so that they could do it when they were at school with their teacher and download it. And so we had some kids who wanted to enroll in the summer who didn't have access. And so we reverted to our usual thing and we just gave them a phone. So we didn't want to exclude anybody. The app features include steps, kilometers, moves, and calories. Moves is the measure that we convert, that's converted and displayed as steps. So moves are the main goal emphasized, and all participants see their own data activity on the app again, or they can see others' activity if they're in the intervention group. So this is the dashboard. It's very, very simple. So it's not hard for the 8-year-old to understand. And the 15-year-olds might be a little bored. They can all, you know, like I said, they can look at it on the computer too. The intervention participants can see others' activity through the platform's challenge feature. And the leaderboard displays the participants' nicknames and how their activity compares to the others on the app. So my staff wear these and exercise so that they compete with the kids on the leaderboard, and they'll send them a text. They can send them a little text through the app or a message through the app to tell them that they're beating them or that they need to try harder, because Sunshine #1, that's Sarah, my study coordinator, is in the lead. I tell her she can't always be in the lead, so take it off. Or again, on the computer. Okay, so this was a study that we did a prehabilitation intervention for children with extremity sarcomas to determine if kids with a malignancy could participate in a preoperative exercise program. So again, the pediatric oncologist in the room can tell you that they're getting some big bad chemo during this time period and they feel like crap. And we wanted to see if we can get them to exercise during this time period. And then we compared them to individuals, historical controls who didn't have this intervention prior to surgery. So our population was mostly male and reported their race as white. 2 of the 14 participants had an amputation and 11 had osteosarcoma. Their mean age at diagnosis was 13. The intervention was feasible, so 12 of the participants, 12 of 14, reported completing at least 50% of their exercise visits with the physical therapist. Although some of the evaluations were very difficult for participants to do because of their tumor location, their surgical status, the fact that they had an IV in, that doing the 9-minute walk test is hard when you have to push an IV pole and you feel like crap. The intervention group, when compared to historical controls, had better mobility 10 to 12 weeks post-surgically, demonstrating some initial efficacy. So thus, if the NIH ever funds me, I have a randomized study plan for this population where the intervention is going to be delivered prior to local control and my outcome is going to be tumor biology. because there's some evidence in the breast cancer literature that if you starve people or if you exercise them, you impact their tumor outcome. Well, I'm not going to starve a child because the dietitians and oncologists won't let me, and that would be too hard, right? But I think that if we can exercise these people, we might be able to impact tumor biology and also potentially immune function. I haven't found an immune function collaborator for that yet, though. But anyway, I'm interested in that. And I submitted this grant not for tumor biology, but for a cardiopulmonary outcome, and got 12th percentile. And I'm a seasoned investigator, so I didn't get funded because you had to be at 9. So I talked to the program officer and he said, maybe we should switch to tumor biology. That was one of our exploratory aims, and the study section might like it better. I'm like, all right, we'll try that next. So this is a study at St. Jude for children with central nervous system tumors, medulloblastoma, and it's finished. It used a hybrid approach. So we started with an in-person intervention because they're a captive audience while they're receiving proton beam radiation. And we did high intensity interval training for 6 weeks. And then we did an additional 4 weeks of the same training at home with the parents. So the parents were supposed to deliver the intervention. So we've completed enrollment, and all 83 participants have completed the intervention and are out to 2 years. The paper's written, but I'm going to show you the results. So the assessment was scheduled for 3 hours. So just to know, if you want to do something like this, they have a physical performance assessment that takes 3 hours, and they have medulloblastoma, but they haven't had chemo yet. So they've had surgery. We wait till they're recovered to start a little bit. And then we break it down into 2 1.5-hour appointments, and make sure that they get a snack before and after each appointment. They get fatigued really quickly. So my staff is really good at this, and we can test kids as young as 3 on the bike now with no problem at all. They're really skilled. We've done 30,000 cardiopulmonary exercise tests in our cohorts since 2006. So the staff in the lab are really good at it. We use a cycle ergometer for kids because they don't naturally run continuously. Children run, stop, run, stop. It's easier to get them to pedal continuously. And also kids with medulloblastoma might have a balance disorder. We don't want them to fall down. And then we do some other motor testing using the Brunitz-Otzkoretsky test of motor performance because I was trained at the University of Minnesota. It's a good test. And the BOT3 just came out, so I got to get new equipment. So the intervention is PT 3 times a week for 60 minutes for 6 weeks, and they spend at least 30 minutes in their target heart rate zone. 55 to 65% of their heart rate reserve, and then any functional activity to maintain their heart rate zone. So if they're a younger kid, we want to make it fun and keep their heart rate up. And if they're an older kid, they're usually willing to do something like ride the cycle or do HIIT training. And the control group got traditional PT, where they tend to mostly work on function and not focus on cardiac outcomes. So these are our results showing a change in peak oxygen uptake in a model adjusted for baseline stratification by cancer risk group, sex, and age. Those in the exercise intervention had a mean increase in peak VO2 of 1.62 milliliters per kilogram per minute, compared to a decrease in the controls of minus 0.49 milliliters per kilogram per minute. So what's interesting about this study is that we had a stopping rule, which we met for efficacy. So we had to stop early. So we think that if we could have kept going, we would have had much larger results, but we had to stop and give everybody the intervention. So now the standard of care at our institution, and hopefully once we publish this paper and maybe do a larger trial, everybody will get aerobic training during medulloblastoma therapy. Might be hard. So we also looked at cognitive function post the intervention, and this shows the association between peak oxygen uptake and both psychomotor function and working memory, which were significant at the end of the intervention. for both groups, actually, looking at the association between peak VO2, which is on the x-axis here, but for the psychomotor task, the kids in the aerobic training group did better than the kids in the control group. So this is an ongoing study using exercise with NAD+ precursor supplementation for adolescent and young adult stem cell transplant survivors, although the youngest participant in this study is 10, so I don't know if I'd call that 10-year-old an adolescent yet. But this study is, the PI of this study is Goli Mustafa Mohab at CHOP, and we enroll patients at CHOP, City of Hope, and St. Jude on this study. My team is primarily, although we do enroll patients at St. Jude, we're primarily responsible for the exercise component of this intervention. So NAD+ works to transfer electrons from one molecule to another to allow cells to generate energy. So this is, to help mitochondrial function. And the supplementation is associated with increased muscle mass in rodents, increased mitochondrial metabolism, and then decreased NAD+ is limits aging in a mouse model. So this is what the study looks like. It's a randomized placebo-controlled trial with a 2x2 factorial design. And again, we deliver the exercise intervention, and we also give we don't know what we're giving in terms of the drug, but because they get a placebo or the NR. And the outcomes are muscle mass, muscle strength, VO2 peak, muscle mitochondrial oxphos capacity. And we test all of that with an MRI. True Niagen is what the drug is. This is what it looks like over the counter. We have to actually get the drug from the IDS at CHOP and a matched placebo. And the exercise intervention is 16 weeks. It's similar to our CARHAB or our home heart health study, except for that in home heart health, we taper the intervention based on a person's ability to uptake the intervention. In this study, we don't taper it. So we see them 3 times a week for all 16 weeks and progressively exercise them. And they have a personalized exercise plan, and we send them a stationary bicycle and weights. So in summary, accelerated aging is prevalent among childhood cancer survivors characterized by disease and deficit accumulation, by physiologic phenotype, by molecular markers of aging, and we have interventions underway to remediate frail health, targeting disease and deficit accumulation, targeting multi-components of the physiologic phenotype, and targeting molecular aging. And also, it's important to know that we're trying to move this towards treatment and survivorship so that we don't develop late effects, or so that we can prevent late effects. So given the data sharing policy, I'd like to tell you that most of the data that I've presented today, except for the intervention studies that haven't yet been published, are available on the St. Jude Cloud for your download and use. So this is just a snapshot of what the cloud looks like without Les's picture. Les's picture is at the top. So those of you who know Les Robeson, his picture is right at the top. But this is what it looks like. And if you go to this survivorship community, you can click on, if you click on View Community, then you can get access to the data. And you can play around in the data too. So there's some, you can do a regression in there, and you can do a t-test, and you can make little graphs and stuff like that. Kind of like SEER. But you can also download the data. So if you want to download the data, the phenotype data, the clinical and laboratory data from St. Jude Life, the questionnaire data from some of the CCSS, not all yet, but it soon will be up there. CCSS cohort is up there. And then the genotype data is there if you have the computer capacity, which at the University of Minnesota, you would have to get the BAM files. They're huge. So downloading them, you can't just download it onto your laptop, or your laptop will not be happy. And then if you click on View Publications and you see a publication, there should be a Zenodo link to the data that's associated with that publication. So I feel like that was my biggest accomplishment in 2023 was to figure out how to share data according to the NIH policy by getting it up here on the cloud so that everybody in the world could check and make sure. So I got you know how you get those emails when you have a paper that says, I'm doing this meta-analysis and blah, 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 and I need access to your data. And I said, here's the link. And I sent them the Zenodo link, and I didn't have to go like dig. So that was kind of beautiful. Anyway, all right, so thank you. And I'm happy to take questions.
I have microphones for anyone that has a question.
That was really interesting, and I some of what you were saying actually took me back 20 some years. I was looking at some old papers that we wrote, like in the early 2000s. We at that time we were focused on cell cycle progression in human immune cells, and so we spent a lot of time looking at T cells. And I'm still wondering. I talk to Paul and Laura all the time about why they're using P16 as a biomarker, because the data, at least the data in the 90s and early 2000s, clearly showed that the senescence marker in human T cells were P18 and P19 and not P16. It's a little bit different from mouse. And this actually led me to think about comorbidities. We were looking at tobacco use and its impact on immune cells. And we found that tobacco use actually increased the senescence phenotypes because it actually was inducing signaling that altered cell cycle progression. And so it was kind of a rewiring of the cell cycle. So I was wondering if you've looked at any comorbidities in the cohort as you're tracking their biological aging and their fragility, if you're looking at things like tobacco use or lean body mass or other things that might actually impact those outcomes and might confound your data.
So yeah, we capture all of the lifestyle variables, and for tobacco use, we also have cotinine levels in blood because people don't tell you the truth about tobacco use. So, and then in terms of lean body mass, we get a DEXA on all of it's St. Jude lifetime cohort. Speaking of now, we get a DEXA on everybody at every visit. So we have lean mass. And so we can look at those things, and some of the data that Dr. Jiaming Wang has published and that Cindy has published will show you that, Cindy uses our cohort all the time, will show you that there are associations with, especially with methylation as an outcome, and not so many studies that have showed associations with P16, and that's very interesting that we should maybe look at other biomarkers. We've looked at some inflammatory markers, and I'm currently having a colleague who's an immunologist take the 6,000 blood samples and do immune profiles so I can take a look, because I'm interested in knowing whether or not there's something going on that we can't figure out. And I said, well, maybe we should just it's kind of like doing a GWAS, just like do a broad sweep and see if there's some sort of a signal for immunity. So he's doing this sort of soma scan type broad immunity in the cohort, and hoping that we can see if we can figure out what's going on. Because there are a subset of people who do a lot worse with their late effects, like with their chronic diseases, like kids with Hodgkin lymphoma, they just do terrible. So it'd be interesting to see.
There is a lot of literature going back. Also the methodology that you can.
Okay, great, thank you.
Thanks for that great talk. So I have a question for you just about kind of building off that. We spend a lot of time talking about what's the right measure for accelerated aging in cancer survivors. You mentioned P16, you mentioned some other markers. There's obviously the physical measurements as well. A lot of our team also looks at different aging clocks, and I'm wondering if you have thoughts about that, because like I said, we spend a lot of time talking about it, and then there's other measures like biologic aging, which take into account things like creatinine and white blood cell count and stuff that, frankly, we order routinely. And if those are comparable, you would think, why are we not building off of that work too?
Right, so last week before I came here, I read Heather's paper on the pace of aging is what I call it. And not me, one of the postdocs at the NCI recently took all of those markers that you're mentioning, all those pace of aging, all the clocks, and it looks like it looks like some of the clocks work well, like the Duden pace of aging clock looks like it works well in cancer survivors. So it's like they're not all the same. And then some of the ones that look at DNA methylation are pretty solid. But that's not a technology that's easy, right? You have to, eventually it'll be $2, but right now it's not cheap or easy, but all the other stuff that you measure is. So I think that we should, figure out what the best measures are and use those clinically. Because I'm always the person in the room, even though I'm an epidemiologist, so I'm into discovery, right? The people in the room who are talking about this, we're going to discover, Cindy's used to me now, so we're going to discover this minute genetic marker of XYZ. I'm like, okay, but what are we going to do with that marker of XYZ? How are we going to apply that for our survivor who's in the clinic? Because not every clinic in rural Arizona, let me give you an example, who's seeing this survivor who can't go back to their cancer center and have a whole genome sequencing is going to be able to measure that marker and understand that they're at risk. And not every cancer center is, I don't know, maybe every pediatric cancer center, and Joe can maybe speak to this, or Lucy, is going to do whole genome sequencing to identify risk for their survivors and put it on their survivorship care plan so they know what to do with that information. So basic clinical markers, I frankly think that everybody should get their hand grip checked and do a 6-minute walk and have the clinician take a good look at what they look like. And then you might not know that they're minute risk for ovarian insufficiency, but you're going to know that from measuring the biology that you measure in the clinic, or their risk for future heart disease. But again, you're going to know that because they got treated with a whole bunch of anthracyclines. So there's good data out there. I think that we need to use what we have. And I think that's one of the new movements, and that's not a new movement, but a new slant in pediatric cancer survivorship is to take the information that's in the guidelines for survivorship care and make them simple enough and pick the best measures that are simple enough so that everybody can use them. So when they're going to see, their primary care pediatrician who's seen one cancer survivor in their entire life in their clinic, somebody doesn't have to read the COG long-term follow-up guidelines, which are excellent, well done, academic, solid, and not very useful for your basic clinician, because they're not going to dig through all that. They might not even remember what an anthracycline is, frankly. So yeah, that's one of the new things. So I think that's a good point. We need to figure out the best markers. And again, I think everybody should get their hand grip strength tested and go for a walk and have the good clinician who's experienced take a look at what they look like and then say this person's at risk for accelerated aging. Let's refer them for some exercise. And once all of the people who are doing senolytic agents figure out which ones are the best, we can give people, what did Laura tell me this morning, metformin, dasatinib, and quercetin together. Joe.
Kiri, I'm not quite sure when we get to the point where we're whole genome sequencing everybody when they come in, but it's going to happen, I'm sure. I had just kind of a different question. You've shown pretty conclusively that you can change the trajectory of strength, of lean mass, things like that. What about sustained effect of exercise? Are you following up on any of these intervention groups and seeing how they differ from controls in 1 year or 2 years or 3 years?
Yeah, so the easiest evidence is our prehab program that we did with the kids with sarcoma. So we're following, there's 14 of them, they're all still alive. They had localized disease. So they're doing much better than in terms of their physical performance than their colleagues. In fact, they were more likely to request additional PT when they got done with therapy because they were doing so well, because we kept them strong during therapy. That's why I want to look at tumor biology, kind of. But I mean, so they're doing very well. And then our strength training program, those are St. Jude Life participants. And we have to they're disqualified from participating in like any observational studies now because they're so much stronger than their colleagues. So we've been following that group of people. There's not many of them yet because they're small studies, but so far we have some other work that we've done with bone health where we put people on a vibrating platform and their bone strength or their bone density is better than, we did it when they were 7 years old and 7 to 14 years old, and now they're in the St. Jude Lifetime cohort, and their bone density is better. I need to write that up. The bone density one is interesting. We put them on a vibrating plate for a year, and their bone density is better. So, and it stayed better. And we didn't do anything else. We did no other interventions with them. Now, some of our survivors who were in our resistance training program became gym rats, which is nice. And so that's a little bit different phenomena. That's health psychology, that if you give people, I was talking about theory this morning with Laura about, I was on a study section that had a lot of behavioral people on it, and they would be not wanting to fund people because they didn't pick their behavioral theory. And I used to be the one who would say, who cares what behavioral theory they picked? If they can get people to exercise, let's just go with it. So I think that, if we can get people to engage and they feel rewarded because they engaged or because they get endorphins or whatever they get. I think somebody needs to study that. I have a postdoc who's super interested in that. So yeah, I think that we're seeing in our small groups, again, I think the first group that will have the longest follow-up was going to be this home heart health study, where we have quite a large number of people, and then the COG study. So if we can keep in contact with all those survivors who are on the COG study, because there's 308 of them, we have like 8 to enroll and we'll be done finally after 7 years. Hopefully those kids are still active. They've been very adherent to their intervention. So I wouldn't say they've been very active, all of them, but they've been very adherent to downloading. So see, that's a good point. You still exercise.
So we have a question from online. Dr. Yee asks, can you say a word about the tumor biology intervention? What are you hoping to affect, and what's the primary endpoint?
The primary endpoint is tumor vasculature. So looking to see if the sarcoma tumor, when it's taken out, has more organized vasculature. So my collaborator for that is Carrie Shadler at MD Anderson. She studies vascular biology in mouse models. So, and she's done some work in pancreatic cancer showing that if you exercise, you improve essentially delivery of the chemotherapy to the tumor by exercising.
Do we have any more questions? All right, well, let's give Dr. Ness a round of applause. Thank you.
Thank you.